control of the cell cycle. Now this stuff is hard.
Oh God.
Just saying.
So you're saying,
Do they still say just saying?
Yeah.
They say cat.
Here's the cell cycle. Let's take a look. Remember G1, what happens in G1?
Growth.
The cell grows and gets bigger and bigger. And it could stop growing completely. What do we call it when it just stops? What does it go to? Kind of goes off to the side, you know what I'm talking about? It just pulls. G0. G0. But one thing the cell can do is it has a way of checking to make sure everything's okay in the cell. These checkpoints are rather complicated. I'll get into a little of how it works today. Yes?
2-day chapter?
No. We aren't going to go into how complex it actually is. I'll give you a little hint of it. But you just have to realize that there's a spot where the cell can kind of check and make sure the DNA is okay, make sure there hasn't been any damage. And we call these checkpoints. There's actually 3 different checkpoints. There's a checkpoint in G1, and then what happens in S phase? Do you remember? DNA has been copied. Now things can go wrong when DNA is copied. Mutations is what we call them. And there's actually about 6 or 7 mutations every time a cell's copied. Most of them are harmless, but some of them can be bad. So there's this S phase where the DNA is copied, and then there's this G2 phase, and there's another checkpoint right there. It checks all the copied DNA and makes sure it was copied correctly. And also checks to make sure the amounts of proteins and such. Remember G2, the cell's making proteins it needs for cell division? We got to make sure that all that's being made, and there's ways that the cell has to do that. So there's a G2 checkpoint. And then there's a checkpoint in mitosis. It's right in the middle of mitosis at metaphase. It's called the M checkpoint. And we check to make sure that all the spindle fibers, Carter, you with me?
Yeah.
Check to make sure that all the spindle fibers are attached to each chromosome. That's basically what happens in the M checkpoint. And so, because if you don't attach a spindle fiber to each chromosome, they won't get pulled apart correctly. And you could end up with too many chromosomes in one cell. So, there's all these checkpoints, 3 checkpoints, G1, G2, and M.
What does G1 check for?
G1 checks the DNA. All of them, all the checkpoints are checking the DNA to make sure there's not a problem.
Does one check to see if the organelles moved out of the outside?
Organelles move.
They move towards the outside. Don't the organelles move?
You talking about the centrosome?
No, they move to the outer part of the cell.
The organelles?
Yeah. Don't they? That's what I said in the last chapter.
The organelles move? If they do, I don't know.
I'll pull it up. Check.
All right. My answer is I don't know. I don't know exactly how they're checking or what they're checking. But you don't have to know either, which is nice.
Ooh.
Now, we do know that the checks involve a couple proteins. One of the proteins is called a cyclin. And the cyclins are proteins that are made, and this is a graph showing the various cyclins in a cell. And let's just take a look at this cyclin E. Cyclin E is started to be created in G1, and its amounts go way up, and then right here at the beginning of the S phase, the cyclins are then destroyed and their numbers go down. And then the cell will go through its phases, and then the next time it's in G1, it'll start making them again. These cyclins help control the cell going through the different phases of the cell cycle. So in other words, when cyclin E peaks, the cell knows it's going to now go into S phase. When cyclin A peaks, the cell can start going into mitosis. You know, G2, you can progress through the stages. The peaking of the cyclins is a signal to the cell to move on through the cell cycle. If, let's say, you didn't have any cyclins, the cell would be stuck in G1. It wouldn't, if it didn't have cyclin E, it wouldn't be able to go on to the S phase. This peak of cyclin E sends it forward, in other words. Now, the other protein you need to know is called a CDK, cyclin-dependent kinase. And they kind of show how these molecules interact with one another. Here's a cyclin molecule. The cyclin molecule will bind to a CDK, cyclin-dependent kinase molecule. Do you all remember what a kinase does? We already studied those. It does what?
It phosphorylates.
Phosphorylates something. It phosphorylates other molecules. So think about this. If something's called a cyclin-dependent kinase, what does that sound like it is? If a kinase phosphorylates other molecules, what would a cyclin-dependent kinase be?
It needs kinase to.
Wait, say it?
It needs cyclin to phosphorylate molecules.
It needs cyclin to phosphorylate other molecules. It's a kinase that needs cyclin. It's dependent on cyclin. So we call these cyclin-dependent kinases CDKs. So, take a look here. So, this cyclin-dependent kinase will bond to a cyclin. Now they form this, they're bonded together, this picture down here. Now there's some protein, we call it a phosphate donating protein, which is a kinase. There's a kinase that will bond to this CDK cyclin complex and drop off a phosphate. So you can see this kinase gave a phosphate to this kinase. Now this kinase, which is the cyclin-dependent kinase, has the phosphate. Now what happens? This CDK cyclin complex, the cyclin-dependent kinase, has got the phosphate here. It's going to give the phosphate to some target protein. And now this target protein breaks away. And here we have the target protein. And it can then cause things to happen in the cell. So it's a domino effect, really. This thing attaches to this thing attaches to that, drops off a phosphate. This thing attaches to the target protein, drops off a phosphate, and now the target protein can do something. Would you like to see video footage?
Yeah.
Video footage. Hey, somebody get the light. Does it still show the rainbow?
Right now it does, but
Sometimes it does, sometimes it doesn't.
Yeah, right now it's good.
Cells grow during the G1 phase, and the genome replicates during the S phase. During the G2 phase, cells continue to grow and prepare for cell division. During the M phase, mitosis occurs. Cytokinesis, C, is the phase when the cytoplasm divides, creating two daughter cells. Three principal checkpoints control the cell cycle in eukaryotes. The G1 checkpoint makes the key decision as to whether the cell should divide, delay division, or enter a resting stage. The G2 checkpoint assesses the success of DNA replication and triggers the start of the mitosis, M phase. If this checkpoint is passed, the cell initiates the beginning of mitosis. The accuracy of mitosis is assessed at the M checkpoint. This checkpoint occurs during metaphase and triggers the exit from mitosis and cytokinesis and the beginning of G1. At the G2 checkpoint, cyclin-dependent kinases, CDKs, phosphorylate histones and proteins that carry the cycle past the checkpoint into mitosis. During G2, the cell gradually accumulates G2 cyclin, also called mitotic cyclin. The cyclin binds to CDK to form a complex called MPF, mitosis promoting factor. When the level of MPF exceeds the threshold necessary to trigger mitosis, the G2 phase ends and mitosis begins. One of many functions of MPF is to activate proteins that destroy cyclin. As mitosis proceeds to the end of metaphase, CDK levels stay relatively constant, but G2 cyclin is degraded, causing progressively less MPF to be available and initiating the events that end mitosis. After mitosis, the gradual accumulation of new cyclins starts the next turn of the cell cycle. The G1 checkpoint is thought to be regulated in a similar fashion. The level of G1 cyclin increases and associates with cyclin-dependent kinase, CDK. Eventually, a threshold ratio that triggers the next round of DNA replication is reached. The cyclin is degraded and the cycle begins again.
So, do you see how it said the cyclins you saw the cyclins interacting with the cyclin-dependent kinases, and then did you see in the video how the cyclins were degraded?
Mm-hmm.
Something was tearing up the cyclins. That's why the levels will go down. So the cell will make these cyclins, and the cyclins will interact with the cyclin-dependent kinases and cause the next phase to happen, and then the cyclins are degraded. And so it cycles. That's why they call them cyclins. They go up and down, and then up and down the next time the cell multiplies, and then up and down. So that's where we get the word cyclin from. Here's one more video.
Cyclins are proteins that regulate the cell cycle. The CDKs are cyclin-dependent proteins that must complex with cyclins in order to act. S-cyclin accumulates and binds CDK, forming the M-phase promoting factor, MPF. MPF activates other enzymes that initiate the signal to synthesize DNA. Now the S-cyclin degrades, and another cyclin, M-cyclin, accumulates. M-cyclin binds CDK, forming a new MPF that activates still other enzymes, initiating the mitosis signal. Finally, the M-cyclin degrades, so that the cycle can start again.
So, this is a molecule called RV. And in this first picture, it's unphosphorylated. So, oh, thank you. This is the unphosphorylated state. This is the phosphorylated state. This is DNA. And this E2F, that's called a transcription factor. Now, we're actually going to learn about transcription and translation later. But this E2F is a molecule that can start transcription of the DNA. And when the DNA is transcribed, that means we'll make a protein. So if this thing is on the DNA, a protein can be made. But right now it's not, because the RB is unphosphorylated. But when one of those cyclin-dependent kinases puts a phosphate or two on the RB, so now the RB has been phosphorylated, then the RB will basically drop off its transcription factor on the DNA. Now the DNA will be transcribed, and basically the DNA is being read. And reading the DNA code can end up making a protein that is involved in cell division. So this is an example of how the cell can be pushed forward through its cell cycle. All you got to do is phosphorylate this RB protein. What phosphorylates it? A cyclin-dependent kinase. Now, here's a picture that's not in your book. Because they think it's too crazy for you.
I see that.
You know what? I might think it's too crazy for me too.
You never know what they're going to ask on the test, but I want to show you how to read something like this. You're not going to have to memorize this or anything like that. But take a look at here how we progress through the cell cycle. Here's G1. There's a little resting phase before S. G1, remember, G1 the cell is growing, and then S, it's copying its DNA. How do we get through G1 and S? Well, we use CDKs and cyclin-dependent kinases. We use cyclins and cyclin-dependent kinases. There's a cyclin, there's a CDK. Remember they came together? Now, all of these other factors are factors that can influence these cyclins and cyclin-dependent kinases. Wherever you see an arrow, here's an arrow here. See that arrow? That means this causes this to happen. An arrow means it's causing it to happen. And this shows the RB and the E2F, the RB dropping off the E2F, just like I showed you in the last slide. And then the RB goes away and the E2F is connected, and we can go through the cell cycle. So there's a lot of other stuff involved. Now when you see, notice this, notice all these. These aren't arrows, they're little lines. You see how this comes down and there's a little line there? That doesn't mean it's causing this to happen. It means it's stopping it. It's inhibiting it. So wherever you see a little line that ends like this, if something's going like that, that means it blocks. It blocks the next thing. If something's going like that, that means it causes the next thing to happen. So these are inhibitors, and these are stimulatory molecules. An arrow causes something to happen, and a line blocks it. So, how can we stop the cell cycle? Let's look way up here. DNA damage. What if something's wrong with the DNA? So, if something's wrong with the DNA, usually there are molecules that come off of the DNA, atoms or molecules. Anyway, the DNA damage stimulates whatever this is, and that thing stimulates a molecule called P53. So often the stimulation is due to a phosphorylation, adding a phosphate. That's often what the stimulation is. I don't know if it's not in every case, but anyway, perhaps this adds a phosphate to that, perhaps this adds a phosphate to that, I'm not sure. But P53 stimulates P21, and then what does P21 do? Follow this line down here. It stops the CDK cyclin complex from pushing the cell onto the next phase. It stops it. So does it make sense that DNA damage stops the cell cycle? That's what it's showing here by this line with that thing. Does that make sense?
Mm-hmm.
Here's growth factor. Growth factor stimulates this thing, which stops this FOX protein from acting. So growth factor is going to stop this FOX protein. What does the FOX protein do? The FOX protein causes this thing to go into action, which inhibits the cell cycle. See that line there? This thing inhibits the cell cycle. The FOX protein causes that, P27, to stop the cell cycle. So what does growth factor do? It stops the FOX from working. If you stop the FOX from working, you don't stop the cell cycle. You keep it going. So the growth factor keeps the cell cycle going. Do you see how complicated it is? How many different factors there are that either stop the cell cycle or start the cell cycle? This is why cancer is such a tough thing. Cancer is when the cell cycle is moving too fast. And one of the upcoming chapters talks about cancer. If the cell cycle is going forward too fast, the cells will start multiplying too fast, and you'll get a lump. We call it a tumor. And too many cells in one place. It could also be a problem if the cell cycle is going too slow, right? You don't get growth of cells that you need. So the cell cycle has to proceed at just the right speed. And there's a lot of molecules that control that. And this is just one part of the cell cycle. This is just G1 and S. Remember, there's G2 and there's mitosis. And G2 and mitosis all have their own stimulatory or inhibitory molecules that can start or stop those phases too. And you got to take into account all of this. Imagine the cell cycle going all the way around, and at every event there's a whole bunch of molecules like this affecting it. Isn't that impressively complex?
Mm-hmm.
The complexity of it is really just impressive. Now, for this class, you're not going to have to understand the whole thing. They'll just ask you about. They'll often give you a figure though with the stopping and the starting, and they'll ask you to interpret. Oh, if this molecule that stops this is mutated, then it's not going to stop it. And you got to think, well, what would happen if it didn't stop it? And that leads us into the next section. I'm going to go ahead and go through this because we're going to start our lab tomorrow. It's going to be, we're making it a 2-day lab instead of one. Question?
Oh, no.
So I'm going to go ahead and talk about cancer a little bit, which is the next section. There are different types of genes that you need to know. One's called a proto-oncogene. Proto-oncogenes move the cell cycle forward. So they often use an analogy that a proto-oncogene is like the gas pedal on a car. for the cell cycle. Imagine the cell cycle is a moving car. Protooncogene is a gene that acts like the gas pedal. It moves the cell cycle forward. If a protooncogene mutates, it becomes what's called an oncogene. If an oncogene is like a gas pedal that's stuck in the down position, you're really moving the cell cycle forward. And that's going to cause cancer. So proto-oncogenes are normal genes that cause the cell cycle to move forward. They can mutate into oncogenes that really speed up the cell cycle and get it going too fast. And so they show the car running into cancer, the car crashing. That's the cancer that's caused when a proto-oncogene becomes an oncogene. By the way, onco means cancer. Have you ever heard of an oncologist?
Mm-hmm.
It's a cancer scientist. So, an onco, you can imagine an oncogene is a cancerous gene. So there are genes in your body, if they mutate, they cause proteins to be made that speed up the cell cycle. All those proteins that are arrows, it'd be like if you increase the arrow somehow, if you made too much of that protein. If you made too much of the proteins that are moving the cell cycle forward, you're going to have problems. You're going to be going too fast through your cell cycle. Now, the other kind of gene is like the brakes of a car. It's called a tumor suppressor gene. Tumor suppressor genes are like the parachute here, or the brakes of the car. They work against proto-oncogenes. The tumor suppressor genes slow down the cell cycle. Whoops, go back. So, P53 is a protein that activates P21, and P21 stops the CDK cyclin complex. So both the gene that makes P53 protein and the gene that makes P21 protein, those are two of them your book talks about, those are both tumor suppressor genes. They stop the cell cycle. They prevent the cell from multiplying. Tumor suppressor genes are good. You need those. You need something to slow down or stop the cell cycle when there's problems. You could imagine, let's say you weren't producing the P53 protein. What would then happen if there was DNA damage? Raise your hand, don't shout it out. Think about it for a second. If there's DNA damage, this is the kind of question they'll ask you on the test. If there's DNA damage, they'll show you a diagram like this. Maybe not show you all of this. Maybe just show you this little line here. There's DNA damage, and there was a mutation where you weren't producing P53. What would happen? Somebody besides Lottie, she answered the last one. You don't produce any P53, and there's DNA damage. What do you think would happen? Chavs?
The P21 wouldn't be produced.
Okay. And then what would be the response?
Then S phase can occur.
That's right. Well, if P21's not produced,
Then nothing else.
It doesn't stop S phase.
It produces more CDK.
so would the cell go through S phase or not?
It would.
Stop.
It would, because the P21.
If there's no P53, so some reason your body's not producing that.
If there's no P53, then there's no P21, and the P21 stops the CDK from binding to the cyclin. So then S phase would go on with DNA mutation.
So if the P53 isn't produced, you would go forward with the cell cycle. Y'all see that?
Mm-hmm.
You would go forward with the cell cycle.
What would it stop?
So P53 is a protein that stops the cell cycle. And so if it wasn't there, DNA damage wouldn't stop the cell cycle. And you would go on with your cell cycle even though there's DNA damage. So you're basically lacking, if there was a mutation in the gene that produced P53, then you have a mutation in a tumor suppressor gene. That can cause cancer. And so these tumor suppressor genes are important to slow the cell cycle. If there's some mutation in it, for some reason you aren't producing P53, that can lead to cancer. A lot of the cancers people have are due to mutations in P53 genes, the gene that causes the cell to make this P53 protein. And that's what this thing's showing. So normal P53, DNA damage will cause P53, and you see the arrow going forward. That means it makes the cell, P53 can make the cell apoptose, can make the cell kill itself. P53 can cause cell cycle arrest. That means it can cause the cell cycle to stop. P53 can cause DNA to repair itself. P53, after the DNA is repaired, then the cell cycle can restart. So P53 can lead to all of these. Here's an example if you have a mutated P53 protein. It doesn't stop the cell cycle. The cell cycle continues. I think I have apoptosis. Can you find apoptosis in here?
It's down at the bottom.
Apoptosis, here we go. Yeah. The CDK cyclin complex, CDK can cause this FOX gene to be, I think your book talks about the FOX gene. I'm not sure how that works though. A cell with abnormal P53 cannot repair damaged DNA and cannot signal apoptosis. You see the CDK causing this FOX gene to signal apoptosis, but I'm not sure how that works, so I'm not going to talk about that. But anyway, apoptosis is a way that the cell, if there's DNA damage, the cell can end up killing itself. And that's important too. So cells need to kill themselves if they've had damage, and that will often happen. The last section of this chapter talks about how prokaryotic cells reproduce. Now, prokaryotic cells divide in what's called binary fission. It's very quick multiplication. And it shows how they do it here. Do you guys remember that prokaryotes have... do my rope. Hold on. You guys remember prokaryotes have a single loop chromosome? This is the chromosome of a prokaryote. And, of course, it's all coiled up in the nucleus, so it doesn't really look like a loop. You know what we call the DNA in the middle of a bacterium? It's not called the nucleus. It's called the nucleoid. But anyway, what it is, it's just a single loop of DNA. And that has to be copied. So, there are proteins, enzymes. One's called DNA polymerase that lands on the DNA and starts copying in one direction. And there's others that will land on the DNA and start copying in the other direction. And they'll eventually go around and copy the whole thing. And you'll have two loops instead of one. The loops then break off from one another. So if you could imagine if I had a second cord here. should have a second cord. That would be a better way to teach it. And then they move to opposite sides of the cell, and the cell pinches in the middle. And that is what they're trying to show here. Here is the original loop of DNA. And in this next picture, now you have the two loops, like if I was holding the two cords. And then we start building, a little cleavage furrow here is coming, so it's pinching in. And it's kind of like plants do also. It builds a new cell wall between them and eventually breaks into two. We call this binary fission. It's a little bit different than mitosis because there's no nuclear membrane to get rid of.
What happens if it has a flagella? Does it just make a new one on the other side?
Yes. So all of the internal organelles. Now, bacteria don't really have organelles, but they have like the ribosomes that are in them and the flagella. All of that can be made by the new cell. So if there was a flagella over here, well, there is instructions on the DNA on how to make a flagella.
Oh, okay.
And so those instructions are read, and the new cell will just make a flagella by reading the DNA instructions. That's true for anything that's inside the cell. There are instructions for anything that's inside the cell on the DNA. So if the cell just gets the DNA, and it'll need some ribosomes too. Now, ribosomes are scattered throughout the cell. There's usually a whole bunch of them. So when the cell divides, some ribosomes end up on the left side, and some ribosomes end on the right side. If the new cell didn't get any ribosomes, then it would be in trouble and probably die. But that's very unlikely to happen because there's so many ribosomes scattered around.
Oh, since mitochondria have their own DNA,
Yes.
do they go through the cell cycles separately?
Yes, they divide themselves just like bacteria do.
Okay.
And so really what they are free-living bacteria inside your cells that can go through a lot of the same things that a bacteria does, and they can divide just like bacteria do. Yes?
In plasmids, they eukaryotes.
Plasmids? Eukaryotic cells don't have plasmids.
Can they get injected into?
I don't think so. I don't think that a eukaryotic cell has the right equipment to read and transcribe and translate a plasmid. It's a great question, though. I'm not sure. But I think there are special things that eukaryotic cells have on their DNA to get them transcribed and translated that plasmids don't. Maybe you could take that information and add things to it. That might be something you can look up. All right. Well, I kind of went through all the last sections. So here's the deal. So you can turn that off.